Yeah. So, hi everyone. Again, I welcome all of you to a Fusion Talk on behalf of Prescience in Silico, and I welcome you all to this talk. Before that, I'll speak few words about Prescience in Silico. At Prescience in Silico, we team up with both industry and academia to tackle challenges in drug discovery and material science. This idea for this Fusion Talk series, it came from our wish to get experts from different fields to share their valuable insights. And I take immense proud to say that we are one of the first Indian companies to create our own drug discovery platform with advanced AI features. Prescience in Silico was founded by academicians who wanted to make AI-powered discoveries in drug and materials accessible to everyone, aiming to redefine the landscape of scientific knowledge. For today's Fusion Talk, we have Professor Suman Chakrabarty with us, who is Associate Professor at the Department of Chemical and Biological Sciences, SN Bose, Kolkata. Dr. Suman Chakrabarty, he has done his B.Sc in chemistry from Presidency College, Kolkata, and MS in 2005 and PhD in 2010 from IISc Bangalore. After that, he has done his postdoc from University of Southern California, USA, followed by his Ramanujan Fellow. He joined CSIR-NCL as a Ramanujan Fellow in Pune, and he's currently working as associate professor at SN Bose. And the title of his talk is Role of Metastable Conformational States in Protein from Function to Drug Discovery. And we'll be looking forward to this talk. And the stage is all yours, Professor Chakrabarty.
Yeah. Thanks, Anil, for the kind introduction. Let me share the screen. Okay, I hope everything is visible. Let me hide
Yes.
the pins. Yeah. Okay, so yeah, thanks again, Prescience in Silico, for giving me this opportunity. Of course, I know a few of you in person since a long time, and it's a great pleasure to be able to talk in this prestigious platform. And as it turns out, I work on a wide range of problems as I'm going to explain to you. But as you can see in the title, there is one aspect of drug discovery as well. And this is where I think it will be very much relevant to the activities of Prescience in Silico. Now, my title may sound a bit technical or complicated to some of you. I just wanted to mention here that primarily I'm going to focus on metastable conformational states, in the sense that normally when we think about structure of a molecule, it can be any molecule, it can be a protein. Normally, when you think about a structure, we think about the lowest energy structure at the global minimum of the energy landscape. When you look at crystal structure, for example, that is just one structure. But many a times, when it comes to biological function, it turns out that other low-lying thermally excited conformations of the protein or other molecules play an important role in the function. So I'll try to highlight a couple of stories in today's talk, where I'd like to showcase that looking at those less populated conformational states, in addition to the so-called native structure, is very important to understand a lot of biological processes, and including what I'd like to showcase towards the end, that how we can leverage those metastable conformational states in the context of drug discovery. So that's the overall theme of today's talk, that role of metastable conformational states in understanding biomolecular function as well as drug discovery. Okay, so many of you may not know me. Just for a quick introduction about what we do in our group. We are a computational biophysical chemistry group, and as the name suggests, I'm a kind of jack of many trades, but master of none probably. So essentially, we use computational tools, then we use ideas from physics like statistical mechanics to solve problems at the interface of biology and chemistry. So it's kind of an interdisciplinary approach. And we use a molecular thermodynamic approach, which is essentially another name of statistical mechanics, where the idea is to connect the structure, interactions, dynamics to the thermodynamics or function of these biomolecules. So that's the kind of the overarching theme. But when it comes to specific systems, our group works on a wide range of problems, starting with solvation, self-assembly, nucleation and growth, phase transition related processes in soft matter related problems, including amphiphiles and polymers and so on. And a major part of our group's focus is on understanding biomolecular recognition, signaling processes, and allosteric. And this is the part that we'll primarily focus on today. We have been doing a lot of work on protein misfolding and aggregation also, and we are actually revisiting some of these problems that we have been working on earlier. And we have also, in collaboration with other experimental groups, we have been working on gas hydrate, CO2 capture, and those kind of problems. So this kind of gives you an overarching view that we work on a wide range of problems, but the underlying theme is essentially this molecular thermodynamics that I'm basically trying to highlight here. And that ideas are quite general. So let me now take you through the ideas behind the methods or computational approaches that we use. So I'd like to spend a little bit on the introduction that I'd like to tell you how we think about the problems that we try to solve, okay? So essentially the tools that we use is molecular dynamic simulation. And I'm sure in this audience, all of you are familiar with what is molecular dynamic simulation. Essentially, if you have a model of how the molecule interacts within itself or with other molecules, essentially you have a model of the interactions, essentially what is called the force field, then you can solve the Newton's equation of motion, and you can actually see the system evolve in the phase space, both in terms of position and velocity, and essentially you can get molecular movements. As you can see here, I have tried to showcase different examples that our group is working on, and you can see a lot of complicated processes are happening, right? Here we can visualize it. Of course, a lot of things are happening. For example, here, some aggregation of some beta amyloid protein or a drug molecule unbinding from a protein, or it could be protein folding, unfolding, or conformational changes. So these are very complicated processes going on, right? And also we are working on this kind of phase transition related problems, as I mentioned, this growth of methane hydrate, crystal nucleation, and so on. But what I'm trying to tell you here, that here we are looking at a very high dimensional data, right? So essentially, if you have an n particle system, you have kind of 3n degrees of freedom, and you are looking at the dynamics or motion at a 3n dimensional space. It's a very complicated and complex data to look at. But how do you make sense out of it? Or how do you calculate some useful things out of it? You could be interested in some structural property, thermodynamics, or kinetics. So you'd like to compute some useful properties that can be connected to experimental results, right? And this is where statistical mechanics comes into the picture. So how do you connect this high dimensional trajectory into some useful, meaningful properties? So this is what we kind of try to do in our group, try to use statistical mechanical theories to make sense out of this kind of dynamical trajectories. Now, I'll just highlight one particular approach in today's talk, and that is about looking at energy landscapes or free energy landscape. So most of you are familiar with this idea of folding funnel, for example, and many of you know that if you allow unfolded state of a protein, for example, you put it in water, you run a simulation, and it will start to fold. And on the right-hand side, I am just, for schematics purposes, I'm visualizing how it is moving on a free energy landscape. And you can see that there are multiple minima and metastable states on this free energy landscape. And finally, this complicated molecule is actually moving on this free energy landscape until it finally reaches the global minimum or the native structure. So this nice video I have taken from, it is generated by Professor Kendall's group in Stony Brook University, but it very nicely visualizes what I'm trying to tell you here. So essentially, you are trying to map high dimensional data where the dynamics is happening on the left-hand side at a very high dimensional space. And when you try to map it on a free energy landscape, it gives us a very nice physical intuition about what is going on in the system. So essentially, we would like to compute such free energy surfaces. And as all of you know from basic physical chemistry textbook, that if you in principle know the free energy landscape for a process, For example, here, for a polymer collapse problem, you can plot the free energy as a function of, say, radius of gyration, which captures the size, effective size of the polymer. And then if you have, say, let's say, multiple minimum in this system, so these are the different states or the, let's say, the conformational states of this system. Now, if you know the delta G, free energy difference between this system, you can know the population ratio of these different states. If you can compute the free energy barrier, for example, but the barrier is actually this gap going from this minimum to that minimum. If you know the free energy barrier, using certain rate theories, you can compute the rate constant. So essentially, if you can compute the free energy surface as a function of some suitable reaction coordinate here, this X reaction coordinate, then in principle, you can get a lot of useful information about the thermodynamics and kinetics of the system. So this is what we try to do. We try to compute the free energy surface of this underlying, essentially the physical or biological processes that is going on. We try to map it to a free energy surface. So there are a couple of technical problems while in computing these free energy surfaces. Some of you may be familiar with it, but just for completeness, I'd like to highlight a few aspects. For example, first problem would be to actually identify this reaction coordinate, right? So as I mentioned earlier, so essentially, let me highlight the problem here in identifying the reaction coordinate. Imagine that you have a molecule as simple as a butane, right? And when you'd like to compute the energy landscape of this, or conformational energy landscape of this molecule, essentially you'd like to look at rotation across this dihedral angle, right? And again, from basic organic chemistry textbook, you know that you can plot energy as a function of this rotational angle or dihedral angle, and you'll be able to see that there are 3 distinct minimum corresponding to each of these specific dihedral angles. For example, there will be a trans conformation, there are 2 symmetric Gauss conformation, and so on. It's all very simple because there is only one dihedral angle, right? So then you can compute the energy as a function of the dihedral angle. But when it comes to proteins, there are hundreds of dihedral angles, right? So essentially, you will be looking at 100-dimensional data. So the problem of identifying a reaction coordinate essentially is a dimensionality reduction problem. So as I mentioned earlier, we have a huge system. For example, for this system, typical MD simulation going on with a protein in water. Let's say there are 10,000 atoms. So there are 3n degrees of freedom, right? Typically, we throw away the water during the analysis, but even then, there are hundreds of degrees of freedom. But you can go to internal degrees of freedom. For example, if you just look at the phi psi angles, it's still, you have, let's say, 40 variables. So essentially, you have a 40-dimensional problem, even if you throw away water and all other degrees of freedom, right? So typically, what do? We identify some physically meaningful parameters or collective variables, people say, or order parameter, right? For example, RMSD, or root mean square deviation, is a very useful order parameter, reaction coordinate. We have seen radius of gyration before. And when you compute, so it's a mapping from a 3n dimensional vector to, let's say, one variable, right? You are doing some kind of a dimension reduction, and you are mapping this 3n dimensional dynamics into a 1-dimensional dynamics. You have a time series data here. From this time series data, you can now compute the probability distribution or histogram. And from this probability distribution, you can see there are multiple peaks that they will correspond to those states. And then you can compute the free energy surface by simple Boltzmann inversion, for example, this formula minus RTLN of the probability distribution, and you get the free energy surface. So in a nutshell, this is the theory of how to compute the free energy surface. You look at the probability distribution of a collective variable of your choice, then you do a Boltzmann inversion. So all said and done. So first problem number 1 is, what is this X, as I mentioned earlier? Now, many times we use physics-inspired or intuition, right? Chemical intuition. When you look at a problem, we know what kind of parameters, geometric parameters we are interested in. For example, here for protein folding or unfolding, RMSD is routinely used as a parameter. But many times, you will not be able to identify a suitable reaction coordinate. So what we'll do when this intuition fails? For example, we'll see some examples later on where, let's say, a loop of a protein is undergoing some conformational change. So it's a disordered thread. Imagine a disordered thread is going from one structure, one arrangement to another arrangement. So which distances you take, which angles you take, it becomes technically very problematic to identify few specific variables, one or two variables, right? So what we do, sometimes we use approaches from data science, inspired from data science. For example, principal component analysis is a very popular and powerful technique for dimensionality reduction, where the idea is, even if you, let's say, you don't know a few distances, you start with a large number of distances, maybe 40 or 50 distances, then you try to find a linear combination of those distances that will maximize the variance. So essentially, you compute this covariance matrix, you diagonalize, and maybe one eigenvector with the highest eigenvalue will correspond to largest variance. So in a nutshell, the idea is essentially you start with a large number of distances and you try to find a combination. Remember, I said collective variable. So we try to find a combination that will maximize my variation. So then I'll say that maybe one or two or five principal components are good enough to capture the overall dynamics or the variance of the system. So this is one approach. Another recently very popular approach is to look at this variable called TICA, which is time structure independent component analysis. There, in contrast to PCA, where PCA was maximizing just the spatial variance in some coordinate space, it was looking at the spatial variation, right? But what TICA says that instead of looking at the spatial variation, we are supposed to look at the slowest variable, like the kinetics now is put into the context. So this example kind of shows you, for example, you have this two free energy minimum. This is the energy landscape. Now, I am interested in this barrier crossing event. So I am actually interested in this Y component, where I would be able to cross from one minimum, go from one minimum to another, crossing a barrier. Now, that would be the slow degrees of freedom. So I would like to compute this barrier, right? But if you do PCA, probably it will give you this largest variation along the x direction, because spatially it is varying more in the x direction, but kinetically it is slower in the y direction. So TICA will be able to capture those slower degrees of freedom. So in today's talk, in two stories, I'm going to use actually one example of PCA, another example of TICA. So that's why I wanted to highlight these two popular methods of dimension reduction. But as it turns out, both of these methods are actually linear projection techniques. This is something to keep in mind, that both of them are trying to find a linear combination. And we have seen in many cases where such linear projections are not very efficient. So many times we need to use certain non-linear projections also. And this is where machine learning comes into the picture. For example, autoencoder we are using nowadays for certain problems. Today, I'm not going to show you any example of using autoencoders, but this is kind of state of the art where people are now trying to use machine learning to come up with this reaction coordinates, right? So if you have a really complicated processes going on, we can resort to modern machine learning techniques, okay? So that kind of summarizes the idea of how to identify the reaction coordinate. Of course, there is another problem of crossing the barrier, right? So let's say I want to go from state A to state B, and if there is a large free energy barrier in going from state A to state B, this is a back of the envelope calculation using this transition state theory expression. You can see, depending on the barrier, as it goes from 5, 10, 15 kcal per mole, here the average time scale of crossing the barriers very rapidly and exponentially grows from, say, nanosecond to microsecond to milliseconds and beyond. So beyond this red line, whenever the barrier crosses this, let's say 10 to 15 mark, now the time scale of the process is going beyond milliseconds, right? And this is where brute force MD simulations will not be useful, because even with best computational facilities, at least available in India, we can probably go to maybe hundreds of microsecond at best, but mostly maybe tens of microsecond, even not hundreds. So this is the barrier for brute force MD simulation for atomistic models, right? And this is where we need to use several enhanced sampling methods. So that one of the most popular method is metadynamics, where you add some bias potential. So you can see here, a normal MD simulation will be stuck in this minimum. And in a metadynamic simulation, you add some bias potential where you push the system from one minimum and then it will flow to the another minimum and so on. So I just wanted to highlight here that there exist a lot of methods using which you can accelerate the sampling of your underlying free energy landscape. And then you can, even with a large barrier, you can actually sample those rare events and compute the underlying free energy surface. Okay. So this kind of, with this kind of longest introduction, I kind of summarize the technical aspects of that, what kind of approaches we use, okay? So now let's come to the problems, the applications that we like to look at, right? So in our group, we are primarily interested in understanding the mechanism of biomolecular recognition and signaling. And it's a very fascinating problem because all your senses, your vision, tasting, smelling, and everything depends on a very complicated protein-protein interaction network. All of you know about this famous protein GPCR. For example, when some external stimulus will come, this GPCR will get activated. And when the GPCR gets activated, it will further communicate this to some other downstream protein, right? So in our cell can be considered like a factory floor where a lot of proteins are continuously talking to each other, passing on information and so on. But what we would like to really understand is how do proteins talk to each other, right? So it's in some sense, it's an anthropomorphic joke that the one protein is saying, I'm smelling coffee, the other protein says, let me inform others. But essentially, we would like to really proteins don't talk like this. So how do we understand how proteins actually communicate with each other? And as it turns out, we can understand this language of biomolecules in terms of this change in the shape, conformations, interactions, and dynamics. So essentially, we come down to this molecular thermodynamics, right? So again, to pinpoint what kind of questions we're interested in, when we say a protein is activated, what does it really mean? What does it mean that a protein is activated? So what happens to its structure or dynamics? Then, once the protein is activated, how does the next protein, the downstream protein, recognizes that the previous guy is activated, right? Because it's like a switch, it has to detect whether the previous switch is on. And then when the next protein senses that my other protein, previous protein is activated, now the second protein now should become activated, right? So essentially, we'd like to have a language, molecular thermodynamics-based language to explain this whole process. So in a couple of slides, I'm just going to show you a very general framework of how to understand these problems, this kind of problems, by looking at the underlying molecular thermodynamics. A simple example will be to understand how do we see the vision process. And many of you probably may know that our vision involves a protein called rhodopsin, which is a membrane protein. And inside this rhodopsin, there is a chromophore called retinal. And this molecule kind of looks like this. Without going into too much of details, all I'm telling you that when light shines on this protein, what happens, this retinal undergoes a cis-trans isomerization, right? So this double bond will rotate, and this rotation will lead to some kind of a mechanical stress in the protein, and the protein now will undergo some kind of a conformational change, and then an ion channel will open up and some ion will pass, and that will be processed as a nerve signal, for example, right? So you can see that there is a cascade of energy transfer going on from one state, one form of energy to another. But the hallmark is why this rotation is happening is essentially this excited state, the photo-excited state has a different energy landscape. So for example, in the ground state for human, it is stable in the 11 cis form. But when you shine light in the photo-excited state, the cis is not the ground state anymore. So it will essentially in the excited state, the dynamics will take it to some other conformation. So the energy landscape is getting perturbed on some external perturbation, right? So this is the idea that we would like to now generalize to explain this molecular thermodynamics of sensing. Now imagine that if I can have this energy landscape as a function of a conformational coordinate. So now we have a protein and we can compute the free energy as a function of some conformational coordinate. And this is the global minimum. So this is the resting state. You can imagine that this is the crystal structure of the protein. But it may so happen that there are other metastable state, this one, which is low lying. So of course, this has a higher delta G. So maybe it is 10% populated in real system, but it will have some population. The first example I'm going to show you later, I'll show you that how this 10% population of this metastable state can play a role in function. But now imagine that I bring in some external perturbation. It can be light, for example, or anything else. Now in the activated state, this free energy landscape is now getting modulated, where now this other minimum or some other state becomes the global minimum. So I was earlier here in the without perturbation. When you bring in the perturbation, now I am in a metastable state. Now I will flow into this minimum, right? And then I'm now in a different conformational state. Now, of course, in this conformational state, this protein may have completely different properties. For example, it can have a higher binding affinity to other proteins or other molecules so that it will bind better with your downstream proteins and so on. So then you remove the perturbation. Now again, it will flow back to the original resting state, right? So you can see this modulation of this free energy landscape on external perturbation. This is kind of hallmark of signaling and sensing action. So there is a stability crossover or switching on perturbation, right? And this kind of summarizes the overall theme that all we do. So now this framework can be used for any kind of perturbation. In our group, we have been working on, for example, light. It can be binding with a ligand, small molecule, peptide, or protein. It can be protonation change. It can be phosphorylation. So you see that there can be a wide range of different kinds of perturbation, but then what all we have to do is compute the free energy landscape with and without the perturbation, and then see that how the free energy landscape is undergoing this modulation, right? So this one slide actually summarizes most of the stories that I'm going to tell you later, okay? So this is the kind of the physical idea or molecular thermodynamic basis of sensing or signaling, that the way we look at, okay? But it's easier said than done. So the problem is, of course, as I mentioned earlier, x equal to what, right? So this conformational coordinate or x. So this is what will vary across the systems and across different proteins, that that specific choice of reaction coordinate will vary, and the specific way of exploring the free energy landscape will change. So this is where we encounter a lot of technical challenges, but for complex problems. But today I'm not really focusing on the technical aspects. So what I'll do now, I'll take you through 3 stories very quickly. Okay, so 2 stories I'll tell very quickly, and maybe the third one with drug discovery a bit longer. So, but all of them will see that it will involve computing this underlying free energy landscape, looking at those metastable states, and also we'll see that how bringing in some external perturbation like binding with ligand can undergo modulation in the free energy landscape. Okay, so first story very quickly. So what is the functional or native state of a protein, right? So we are familiar with this kind of protein folding funnel landscape where the native state or native structure appears at the bottom. But you can see that in this energy landscape, there could be low-lying metastable states or other conformational states, right? So what I'm going to show you that, so the native state, it may not be just the one structure or the native state, but we should look at an ensemble of structures where there could be many conformations at equilibrium with each other. So some of these low-lying metastable states or the conformational states can be important for function. So one such example is this toxin protein, HEAP A, E. coli toxin protein. I'll not go into too much of the biology, but I would like to highlight the physical aspects. So what happens is in the resting state of this protein, there is this loop. And in this loop, there is a serine residue, right? S150. As it turns out, this serine loops actually serine residue gets phosphorylated by some other protein. And when you compare the crystal structures, in the wild type, this serine is buried inside, right? But if we look at the crystal structure of the phosphorylated state, it is actually exposed outside. So naturally, the question comes that in the wild type or in the native structure, if this loop is buried or if the serine is buried inside, how will it get phosphorylated? Because to participate in the phosphorylation reaction, it has to be exposed to the other protein or water in some sense, right? So the hypothesis is actually the wild type, the buried state is not phosphorylation competent, but it is in dynamic equilibrium with some partially exposed conformation where the serine will be exposed, but that will be phosphorylation competent. And that state will further drive the phosphorylation process. So we expect that there will be some excited state of intermediate that will drive the reaction, but not the native structure. Okay. So to look at this problem, we computed the underlying free energy landscape. This one kind of shows the free energy landscape, and this has been done in the principal component space, PC1 and PC2. And all the blue patches that you see, those are the minimum in the free energy landscape. So all the minimum, all these basins will dictate or correspond to unique or distinct conformational states of this protein, right? And as you can see here, this star, pink star, marks the wild type structure. So, and this red star, that marks the phosphorylated state. So the protein actually goes from this conformation to this conformation. And on this side, we actually show in the same conformational landscape the extent of water exposure. So the blue color indicates how exposed it is to water. So the average number of water. So the darker blue will mean that it is more exposed to water. So you can see the wild type conformation, the buried state was not exposed, more white. And along the pathway, there are many other conformational states which are more exposed to water. For example, the C conformational states, right? So we have also characterized, we have characterized all these intermediate conformational states, but will not go through all the details. All I'm trying to tell you here is this partially solvent exposed metastable conformational states are phosphorylation competent, but not the native structure. And we have proved this by computing this underlying free energy landscape and correlating with the extent of solvation exposure. So what we summarize here is this kind of transient surface exposure may be a general mechanism of phosphorylation of buried residues. And a more generalized conclusion would be that to explain, sometimes we give too much importance on the native structure or the crystal structure. But many times when you try to understand a biological function, maybe that crystal structure is not enough to look at. So there could be other localized conformational changes, those kind of less populated conformational states that actually may play a very important role in the biomolecular function. So all of us should be really aware and appreciate this fact that this conformational, looking at the ensemble of conformation is more important than looking at a specific native structure or crystal structure. So this work has been done in collaboration with Professor Gautam Basu in Bose Institute. So now, very quickly, a second story. Again, here I would like to highlight this fascinating way these proteins can adapt their conformations and how it can play an important role in the biomolecular function. So this work again was done in collaboration with Professor Ranjit Bishwas in our center, Bose Center. They had a very interesting observation that this enzyme lacase, this is a very nice enzyme that can degrade a wide range of dye molecules. And actually this enzyme is very generic in nature. So what they found that this enzyme can degrade these molecules. You can see on this right, these are essentially different organic dyes routinely used in spectroscopy. But you can see that these molecules they vary widely in their size and shape. Some are smaller, some are bigger. And not only that, their charges vary. So here its charge is +2, +1, +1, -1, even -2. So the charge is varying from -1 to -2 to +2, right? So this enzyme somehow it has a substrate promiscuity that it can actually bind and degrade a wide range of substrates. So we'd like to understand how. So first of all, how these different kinds of substrates they're actually binding to this enzyme and how the enzyme is able to actually work on this wide range of substrates, right? So then again, we looked at this binding site of this enzyme. So there is a loop that covers this active site of this enzyme. It's actually multi-copper enzyme. I'll not again go into the details of the enzyme activity, but there is a multi-copper active site. And it turns out that there is a loop that covers this active site and that undergoes a conformational change drastically. So depending on what dye molecule is bound, so you can see this red is the crystal structure, then there is a brilliant blue, crystal violet, and all these are these dye molecules. Depending on which dye molecule is bound, it can adaptively change the conformation or shape, right? So it is like there's a high degree of plasticity. So the binding site is actually changing its conformation and shape to accommodate a wide range of different kinds of dye molecules. And we have computed the binding energy of all these different dye molecules using a kind of a rudimentary MMGBSA kind of method. Of course, there are better ways of calculating the binding energy. But even within this approximate method, we see that the total binding energy is actually very comparable. It's around, let's say, -30 kcal per mole. But interestingly, if you look at the individual contributions, electrostatic, van der Waals, solvation, and so on, they vary dramatically. See, in some of them, the electrostatic is positive, some of them electrostatic is negative, and it's the other way around. So there is a internal cancellation going on between different types of interaction energy, but the overall binding energy is comparable. So somehow this enzyme active site has this ability to accommodate different types of molecules. So for some molecules, it is stabilizing through electrostatic interaction. For some molecules, it is stabilizing through van der Waals interaction, and so on. So this kind of internal cancellation is leading to kind of comparable binding affinity across all of them. Okay, so this actually explains that why this wide range of molecules can actually kind of have a comparable binding affinity across all the molecules. Okay, so for the sake of time, I will just skip through all of this. I wanted to just tell you that we actually tried to characterize this conformational landscape of this binding site. And again, as you can realize, this loop has a very disordered structure and all. So how do you kind of identify the reaction coordinate? And for this, we have identified 38 different distances, then computed principal component analysis. We have performed the PCA, and then when we projected on the PCA space, it shows that indeed the conformation are distinctly different for all the different ligand dye-bound structures. Okay, so I'll not, again, these are preliminary results, and we'd like to actually, we are now working on identifying whether it is an induced fit mechanism or conformation selection mechanism. As of now, preliminary results indicate that it is actually conformation selection, because same conformational states exist even without the dye. So if you just delete the dye, even the apoprotein can exist in all these different distinct conformational states. So again, this is in future. We are now trying to use some enhanced sampling methods to actually compute the free energy landscape of this system for different dye molecule to clearly understand whether this is induced fit or conformation selection. So this is for future. Now, in remaining 10 to 15 minutes, I'll now focus on the major interest of today's topic, and maybe most of you would be interested in the context of drug discovery. So we have been interested in allosteric for a while, and as it turns out that identifying allosteric drugs is a very lucrative strategy in pharma companies, and I'm going to come to that very soon. So our goal would be essentially to come up with allosteric inhibitors or allosteric drugs for inhibition of protein-protein interactions, PPIs, okay? And then the question is, then can we exploit this kind of metastable states and this conformational population shift to come up with a strategy, a rational strategy for designing these allosteric inhibitors and allosteric drugs, okay? So now, a very quick introduction. All of you know how medicines and drugs work, but still, very quickly, I would like to highlight that most of the medicines that we take, they are essentially competitive inhibitors. So in a normal enzyme or protein, for example, this is the active site, and where the substrate will bind and lead to product. So normally we try to come up with molecules that will bind to the same active site, but with higher affinity, right? So then your inhibitor molecule will preferentially go and bind. It will compete with the natural substrate, and then it will bind to there preferentially, and then it will not allow the natural substrate to bind, and hence it will act as an inhibitor. So this is the most commonly used strategy used in your virtual screening and talking and so on. You try to find a molecule with a higher affinity compared to the natural substrate. But now there is another strategy where the inhibitor may bind somewhere else. For example, here there is another pocket somewhere else, which we'll call the allosteric site or distal site. If the molecule binds in this allosteric site, the conformation may change at the active site. See, here it was triangular and here it is some other shape. And then now that the shape has changed at the active site, now the natural substrate cannot bind here anymore. There could be other mechanism, but this is the idea that now your inhibitor should bind somewhere else, and then from a distance, it will modulate the active site such that the protein cannot perform its function as intended, right? So this essentially, this is what is called allosteric, which is nothing but a remote control action at a distance. So we would like to come up with a strategy of identifying such allosteric sites. And why it is very lucrative? Because in many cases, competitive inhibitors can actually have a lot of side effects. Because, for example, for cases like, for example, where ATP is the substrate, and you try to find an analog for ATP, which is a competitive inhibitor, that may compete with many targets, right? But in general, it is thought that allosteric inhibitors are more specific, more selective to a specific protein. So the chances of having side effects or off-target binding would be probably less. So this is something seems to be very lucrative. But what are the problems or the challenges? So the first challenge is identification of the allosteric site. So what are the ideas? You need to identify a distal site, which should be far away from your active site. But more importantly, it has to be allosterically connected to the orthosteric site or the active site. So there has to be some kind of a thermodynamic coupling that binding at one site should alter the binding at another site, right? So there we should identify a distal site which is allosterically connected to the orthosteric site. And next problem is many times such allosteric sites are called cryptic. What do I mean by cryptic? That those sites can be metastable and not significantly populated. What do I mean by that is that this pocket may not be visible or available in the crystal structure itself, because protein is a dynamic system, as I'm going to show you in a movie in the next slide. So the protein is actually a very liquid and flexible thing. Many times such pockets may open up dynamically, transiently, and then those transient or dynamic pockets are called actually cryptic pockets. And so to identify them, of course, now you have to resort to molecular dynamics. Just by looking at crystal structure won't be enough. So how do we identify such cryptic pockets? And then how do we ensure that those are the allosteric sites? So these are the kind of technical challenges that we try to address. I mean, we are trying to come up with some methods to actually resolve this. So essentially, again, this is a movie taken from D. Shaw's group. All of you know that they have this big Anton supercomputer. And I find this movie really fascinating because actually it shows that when this ligand molecule is actually exploding the protein surface, it is actually like a liquid, right? And when it binds to the designated pocket, these blue regions, you see that it is like acting like a clamp. So you probably did not have these clamps in the crystal structure. And essentially, it is like finding keys for a dynamic lock. So when you'd like to design a drug, we don't even know that which conformation I'm looking for as the target, right? So this is the challenge of finding allosteric drugs and also identifying those cryptic sites. So essentially, you're trying to find keys for a dynamic lock. Okay, so the specific system that we are working on, that we would like to come up with some alternative strategy of to treat hypercholesterolemia. Like, I mean, normally many of us probably need to take medicines like statins to lower our cholesterol level, but people have been thinking about other alternative strategies. So I would like to just quickly take you through what is the target here. So in our hepatocyte, there are these receptor proteins called LDLR. So these LDLRs are LDL receptors which bind the LDL particles, right? These low density lipoproteins. And when they bind to these LDL particles, it goes inside these endosomes and it decomposes the LDL, and the LDLR is recycled. So this process actually is essential for continual consumption of LDL that will reduce the LDL level in our blood. But there is another protein called PCSK9. If that binds to LDLR, then this whole complex, you see it's a now ternary complex, this whole complex gets decomposed in the lysosome. So of course, now LDLR is not recycled back to the hepatocyte surface. So this LDL consumption pathway is hindered. So the take-home message is that this PCSK9 LDLR interaction, this protein-protein interaction, it suppresses the recycling of LDLR and it diminishes the uptake of LDL, right? So we would like to block or inhibit this PCSK9 LDLR interaction. So take-home message is we have a protein-protein interaction, PCSK9 LDLR, that we would like to inhibit. And there have been a lot of attempts already, a lot of genetic data available to actually confirm that indeed PCSK9 is a valid therapeutic target for reducing the LDL level. But what is the problem? Why not just competitive inhibitors? In general, for protein-protein interactions, this protein-protein surface has a very large area. So it is very difficult to have a small molecule inhibitor to inhibit PPIs, right? Because you have a large area to inhibit. There are already some surface-limiting peptides or antibodies available in the market, but it is difficult to administer them. You have to inject them. So it's expensive, it's difficult to administer. So the dream would be to have small molecule drugs which are easier to administer and cost effective. So of course, the answer would be possibly to find allosteric sites. So this is the kind of crystal structure. This is PCSK9, which is a multi-domain protein. And this is where the LDLR binds. And so we'd like to find some allosteric site somewhere else, where if you bind a small molecule somewhere else, it will allosterically modulate this binding affinity, and I would like to inhibit this. So that's the goal or idea. So how do we go about it? So first, what we identified is that we first run two sets of very long simulations, and we essentially with and without this LDLR. So essentially one with bound to your protein, receptor protein, and then without that, the apoform of PCSK9. And when you compared these two proteins with and without LDLR, then we identified that there are certain regions on the protein, including today we are going to focus on this loop region, 211-222. This undergoes a large conformational change. So in the apoform, without LDLR, we will call this conf A in the apoform, and conf B, which is populated in the bound form. So what we report here is, if you with and without this LDLR, this loop is undergoing a conformational change from apo to the bound state. So it seems that LDLR binding is leading to this, modulating this distal loop conformation. So it is somewhat allosteric. So binding with LDLR is modulating this the conformational change. So there is an allostery induced by the LDLR binding. So to actually now we would like to characterize this in terms of the free energy landscape. I'll skip this technicality. So what we do is we build something called a Markov state model to characterize this complicated conformational space and compute the free energy landscape. So if there are questions about Markov state model, we can take it up later. But all I'm trying to tell you here that essentially we have computed the conformational states and their population and also their interconversion kinetics. And this kind of summarizes the free energy landscape and also the conformational states. And you can see that for the apo state, here this conformation A is more populated, conformation B is less populated. As you've seen earlier, conf A was for the apo-specific and conf B was the bound-specific. And also there are certain intermediate conformations as well, IM1 and IM2. And when it comes to the LDLR bound state, now you can see the conf B is more populated, conf A is less populated. So through free energy landscape, we have confirmed that indeed that LDLR binding is modulating this conformational free energy landscape allosterically, right? So this is through careful MSM building we have proved this. So it is now proved that LDLR binding modulates this distal loop conformation allosterically. But we would like to do it we are interested in the reverse process. Can it be utilized the other way around? What do I mean by that? That LDLR binding is leading to conformational change. But now, can I change the conformation in that loop that will reduce my binding affinity? So I'm looking at now the reversibility. So whether a conformational change can lead to change in the binding affinity, the reverse process. So we have devised a simple thermodynamic cycle here. I'll quickly take you through this thermodynamic cycle that establishes this bidirectional nature of allostery. So we are essentially trying to look at the coupling between conformational free energy versus binding free energy. So in this protein, this is the binding site. This red triangle is my substrate. It will bind here. So this is the LDLR. And this is the allosteric site that can undergo this conformational change from the square to the triangular state. Now, there are four free energies here. This is the free energy of binding. See, the ligand is binding here when the allosteric site is in the square conformation. So delta G be binding with square conformation. But similarly, your allosteric site can be in the triangular conformation. Then when it is binding, it is delta G binding with triangular conformation. So what we are postulating here, that these two binding energies are different depending on when the conformations are different. Now we can now look at just the conformational free energy. So in the apo state without ligand, this is the conformational free energy of going from square to triangle. And here in the hollow state or the bound state, the conformational free energy of going from S to T. So when you close this thermodynamic cycle, it can be shown that the difference between these two binding free energy, so this binding free energy is related to difference between these two binding delta G, right? Okay. So all I'm trying to tell you here, it might look a bit complicated, but all I'm trying to tell you here that if the ligand binding actually changes this relative free energy, you see that this is the relative free energy of between S to T, that which conformation is more stable. If ligand binding changes the conformational population at allosteric site, it will ensure that that difference will be added to the binding free energy as well. So this difference will be directly related to the change in the binding free energy. So this thermodynamic cycle actually ensures and proves that coupling between the conformational free energy at the allosteric site and the binding free energy. So the strategy would be, or the take-home message is, the binding affinity will be lower for the conformation that has higher population in the apo state. So the apo-specific conformation, so the conf A that was more populated in the apo state, that should have lower binding affinity. Okay, so this is something we have proved through binding energy calculation. We have taken all those conformation structures, computed the binding energy using PBSA method, and we have indeed shown that for the conformation A, the binding energy is much lower, minus 21, compared to conf B, where it is much more negative, right? So you can see that as you go to more apo-specific conformation, the binding energy is becoming less and less negative, right? So, and the binding energy follows the same order as the conformational population. So the take-home, the design strategies for identifying allosteric drugs or allosteric sites, the apo-like conformations correspond to lower binding affinity. So the again, take-home message is, we'll have to compare the conformational ensemble of the, we take with and without your partner protein, LDLR, you compare the apo and bound ensemble, you identify which conformations are unique or more populated in the apo ensemble, and those conformations are guaranteed to have lower binding affinity with your binding protein. And this is proved by thermodynamics. So this kind of summarizes our strategy based on this kind of this bidirectionality. We are leveraging this bidirectional nature of allostery to come up with this strategy. So again, I will not talk about all this, but I just wanted to flash it here that I did not explain how allostery works. And in our group, we have been working on explaining the mechanism, that how the information on one site actually propagates to the other site. And this, in these two work, we have shown that there are pairwise interactions between all these amino acids, and this pairwise interaction, short-range rearrangement can lead to long-range communication. So there are many hydrogen bonding or short-range in contacts. Their rearrangement can lead to overall long-range communication. And in various examples, we have found this kind of thing, that long-range communication can occur through short-range rearrangement. So with that, let me summarize what I tried to tell in the last major story, that it seems that even some structuredless or disordered loop regions may be potential targets for allosteric drugs if they undergo this kind of conformational change on substrate binding. And this leveraging bidirectional nature of allostery, if the substrate binding leads to conformational change, then we should preferentially target those conformations. Those are apospecific, and so to modulate the substrate binding affinity. So this kind of summarizes our rational strategy of identifying the allosteric sites. So with that, let me acknowledge the people who are involved in all these stories. So for collaborations, I thank Professor Gautam Basu from Bose Institute for the first story involving that cholera toxin protein, Professor Ranjit Vishwas from SN Bose Center for the lackase work, and Dr. Salim Shah from Surface Pharmaceuticals, who kind of partially sponsored the last project. They actually brought the problem to us about this PCSK9 LDLR interaction. For funding, I thank Surface, the pharma company, ACRB, DST, and of course SN Bose Center for the funding and the computational facility. The first work and the third work has been done by my PhD student Krishnendu, and the lackase work has been done by Sudipto, who is a student of Professor Ranjit Vishwas, and we are collaborating on this lackase problem. So with that, I thank all of you for your attention, and I'll be happy to take any questions. Thank you. So I think I can stop sharing now so that I'll be able to see all of you. I think, Anil, I cannot hear you.
Yeah. Can you hear me now?
Yes, I can hear you now.
Great. Yeah, thank you for the wonderful talk. Yeah, now we'll take questions. Yeah, so there's a question from Professor Jamdede. Yeah, and he asked, if metastable states were characters in a movie, what role would they play and why? Wow, interesting.
Well, you can correct in the context of the way we analyze in MD simulation. you can characterize metastable states as the actors who get less screen time, for example. So your main characters will get more screen time. So think in terms of population. So in our case, when you compute this free energy landscape, essentially more populated states are essentially the deeper minima of your free energy landscape, and the shallow minimum or the higher lying states have less population. So if you think about a movie, people who come for less amount of time, but probably they have a very important part in the movie. So they come for a small amount of time, but they initiate a part of the story that is, I mean, they do something that is very important for the story. That's the analogy I can draw here.
Okay. Okay, so meanwhile the questions are coming, I would take the opportunity to ask questions.
Yeah, sure.
So let's say for this phosphorylation work that you have done, the serine phosphorylation. So, I mean, is there any, NMR data available just to validate,
This question we got from our reviewers as well.
Okay.
Unfortunately, so we have been collaborating with Professor Gautam Basu, as I mentioned. So, but the experimental part has been done by them. It was not possible to do solution NMR. Of course, it would be ideal to do it. We did not have that facility or the access to that. So what they have done is they have done some protein folding experiments to look at the kinetics, and they have fitted to a statistical model to identify how many conformational states are there. So they could identify that there is some intermediate state, and that population can change depending on the condition. So essentially, from experiment, they had some indirect clue that probably there is some intermediate, but they did not have any direct conclusion from the experiment. And that is where we come in, that through MD simulations, we could prove that. So it is the other way around, that simulation is proving that there exists an intermediate state. But you are right that ideally a solution NMR would be the way to go. But unfortunately, we have not done that.
There is a question from Mahesh. During the binding of small drugs like compound, how does the translational energy get dissipated? And are there energy transmission networks within the protein structure?
Right. So actually, this is an interesting question and part of ongoing research. The energy, you see, as I mentioned already, that in all these signaling processes, there are different forms of energy interconverting. For example, in the rhodopsin, first there is a photoexcitation. Now the light energy is getting converted into mechanical energy, kind of, or chemical energy. Then that is again converted into electrochemical gradient in terms of let's say, proton gradient, and so on and so forth. So that is one way of converting energy from one form of energy to another. So again, to give more precise example, light shining is changing the conformation of the molecule. That will change the dipole moment of the molecule. Like the dipole, there is an NH bond dipole that rotates, and that is actually rotating a dipole. That is actually changing now the electrostatic interaction with something else. So these changes in the molecular conformations are changing the way these molecules interact, and that is leading to like cascade of this energy flow, right? That is one way of looking at it. But actually, there are more complicated ways that is not completely understood. These proteins have these vibrational modes, and there are speculations that maybe there are these vibrational modes which are coupled to each other. So there could be more direct way of having these energy transfers. But to properly look at these modes, one needs quantum mechanical calculations to actually do a thorough job. And it is very difficult computationally to do a full level quantum chemical calculations for proteins. So there are people who are actually looking at this kind of quantum mechanical factors, which may play a direct role in this kind of function. We are looking at it from a classical mechanical point of view. And that's even within that framework, we can explain many of these processes. And as you asked generally how the energy are dissipated, part of it would be thermal energy mostly. That even in a simple reaction, if you think of a simple chemical reaction, A plus B going to C plus D, there is a delta G, delta H, enthalpy of the reaction. And normally that enthalpy will be released as heat. But again, the speculation would be the nature probably would not like to waste many of this energy just in terms of heat. Can it directly internally use part of this energy to do something else? But this is very difficult to prove. And actually, we are trying to do this kind of thing to prove that energy will be actually used to quickly convert to some other form of energy and not to lose in terms of heat. the crudest form would be to just to dissipate in terms of heat. Yeah.
Right. Next question is from Chan Singh. He says, great talk. Can you share your thoughts on accuracy or precision of free energy methods employed in this work versus metadynamics?
Right. Yeah. So, well, I would argue that even metadynamics has its own limitations in the context of convergence and so on. So, what is proven that this kind of, there are different aspects when you choose a specific method for calculating free energy. If you are interested in computing the absolute free energy of binding, yes, one has to use more rigorous methods. But if we are more interested in the relative binding energy, that we would like to order that this is higher binding energy or less binding energy. So even in the context of, for example, binding of small molecules to proteins, it has been shown that MMP-BSA correlates very well with the experimental binding energy in the context of giving the relative free energy that you want to compare between two molecules. So for that, it is reasonably good enough qualitatively. So we wanted to have some quick and dirty way of comparing between different molecules. But if you want to compute the absolute free energy, then of course one has to use, there are many methods. For example, people use free energy perturbation, thermodynamic integration kind of methods to calculate absolute binding free energy. Metadynamics will have a problem of identifying metadynamics or umbrella sampling. They require a binding coordinate, a reaction coordinate. So you have to know the pathway of binding, unbinding as well. Of course, that gives you more information. You get to know the transition states or the mechanism of the process and so on. But yes, so there are, each method has its own strength and limitations, and it will require significantly more computational power to get accurate free energy using metadynamics. So since we are more interested in the relative binding energy, we were reasonably happy with MMP-BSA.
Okay, so now we have question from Shivam. He says, thanks for interesting talk. His question is, if the allosteric is dynamic or entropic in nature and it doesn't show significant structural changes, then how can we capture the binding energetics from their free energy profile, as we won't be seeing much change in their energy profiles? Can free energy profiling will be significant approach to target such kind of problems?
Okay, so again, there are very interesting and important question. So I'll give you two answers. First of all, if you calculate free energy, that includes both energy and entropy. So in principle, even if there are entropic changes, that will reflect in the free energy surface. That is answer number one. So free energy actually captures both. Now, to give another way of looking at the same problem, when you say that if there is in the context of dynamic allostery, people have been saying that in dynamic allostery, there is no change in the structure. It is only dynamics and entropy. But we have shown that actually, when you say there is no change in the structure, you are probably looking at just the crystal structure. But as I mentioned, one should look at the ensemble of structures and the population. So it is not about just structure A, structure 1, and so on. So there is a population of structure A, B, C, D. So the population will mean, let's say, 80% and 20% versus 60% to 40%. So if you look at one structure, you will get only the most populated structure. Maybe it is 80% or 60%, you are seeing only one structure. But actually, this underlying change in the population, that is what is actually important for explaining allostery. So one has to invoke this language of conformational population and how this population is shifting. And that will incorporate both the entropic effect and the enthalpic effect as well. And in some of our work on dynamic allostery that I highlighted, we showed that even for these classic examples of a dynamic allostery like PDZ domain, actually at a side chain level, there is a conformational change, that this side chains reorient. So certain, let's say one set of hydrogen bonds in the apoform, another set of hydrogen bonds in the bound form, and this population undergoes a shift. But if you look at a backbone level, probably you don't see the structural change. But always there will be structural change in the context of population. That will be always there. If two populations are identical, of course they are same. Even the free energy will be same. You cannot say they are different. Even the entropic change means that there is a change of the population. This is something one should keep in mind.
Yeah, the next question is from Ashish Kumar. He says, apart from MD simulations, is there any other method for identification of cryptic pockets? And how can we validate that it is playing a role as cryptic pocket or not?
Okay, so the first definition of cryptic pocket is that it is not visible in the crystal structure. Okay, so there could be many other than MD simulation. Of course, there is Monte Carlo, but I'm keeping it aside because that's also a simulation technique. But without simulations, there would be two ways. One is, for example, the solution NMR we mentioned. So again, the point is you have to invoke dynamics in certain way. So in solution NMR, people can look at other conformational states. It is probably possible to look at other conformational state using NMR, but again, there is a time scale issue. So NMR, certain NMR techniques have a certain time scale window where they can look at. So if the cryptic site opening is not accessible within that time scale, even NMR may miss it. Now, computationally, people are trying to kind of modify this AlphaFold. You know, I think all of you have heard of this AlphaFold, where they look at this sequence alignment data to come up with the structure. Now, people are trying to give it the limited MSA, the sequence data, and playing with it so that we can full AlphaFold to give alternative structures. So right now, you are trying to ask AlphaFold to give me the native structure or the crystal structure. But somehow, if you can modify the algorithm, you can ask AlphaFold to give me some alternative structure, and it's possible that in those alternative structure, there could be some other pocket open, and those could be your cryptic pocket. And also, people are in machine learning community, people are coming up with other kind of generative models where you can directly build a protein conformation using generative models, right? Even in drug discovery, people are using generative models to come up with new molecules. Similarly, you can use generative model to come up with a new conformation. So, but using that, you still get just a pocket. But to prove that that pocket is allosteric, somehow then you have to use either experimental validation or MD simulation to prove that these two sites are thermodynamically connected. Otherwise, just because there is a pocket, you don't know whether that is allosteric. Okay, so that's all I can say.
The next question is from Raju. He says that TICA analysis method always, is it true that TICA analysis method always gives you the better thermodynamics and kinetics together, although we know that it is a linear combination of order parameters?
The short answer would be actually no, unfortunately. So see, always I say this kind of quote that, see, we are doing projections, right? So essentially we are going from a true data is 3n dimensional. And from there, we are doing some kind of projection to a lower dimensional space. So from 1000, I'm going to 10 or 2. Now, there is a saying that all projections are wrong, but some are useful. So whatever you do, you are losing data. Okay. So how much you are losing and how much how much accurate it is, it will depend on the problem at hand. Now, I will give you the other way of looking at it. Depending on there is also compressibility. So let's say in your 3-dimensional data, there is indeed only one collective motion that is important for your system. Then it is enough to just project to one variable. But let's say in some systems, it is not enough to project to one, and you need 10 variables. So even when you're doing TICA, essentially you're choosing how many eigenvectors to keep, right? So you're ordering that I'll take top 2, top 3, top 5, top 10. So if you keep enough of this top n eigenvectors, you will retain most of the information. But how many vectors you need to retain, that is a technical challenge. And then maybe for some problems, it will not be practical. So it may so happen you did TICA, but it turns out that maybe 20 top TICA components are important. So this kind of analysis one has to do carefully. Many times we just choose to just take top 2, but that may be erroneous in many cases. That you'll miss out many of the important degrees of freedom if you just decide to take top 2. And this is where this variational autoencoder and other machine learning based nonlinear techniques, they can have better compressibility sometimes. That with fewer degrees of freedom, you can capture maybe most of the important processes that is going on. So it is all about that what you are trying to look at, and then how many projection variables are good enough to capture that process. So it very much problem dependent.
Okay. Now I will ask few questions. Regarding this allosteric part, the later part of your talk, which was majorly focusing on designing, peptide-based inhibitors.
Right.
So yeah, so did you try using this RF diffusion? Because like recently I was going to
Right.
literature and found this RF diffusion. Could you please comment on that?
Sure. So for the allosteric inhibitor, as of now, our focus was on identifying the site. So see, to begin with, the problem with allosteric inhibitor is that I don't even know where is my pocket.
Yeah.
So because for normal active site, you don't know where, you know where it is from your crystal structure. But first, in this problem, our goal was to first have come up with a strategy of identifying the site, allosteric hotspot or pocket. Then once we identify the pocket, then we come up with the inhibitor. So in our work, as of now, we have not designed the inhibitor per se.
Mm-hmm.
But we did a proof of concept that we have how to select the pocket, and also in that pocket, different conformation can have different pockets. So it is not again enough to look at just one conformation, because there is a conformation population and each of them are different. Now, once we identify which conformation to target, then in that conformation, now we can use either standard virtual screening to come up with new molecules, or we can use this kind of generative models like RF diffusion and other algorithms. And that would be like the future, where we try to design the inhibitor. So as of now, the focus was on identifying the site.
I see. And can there be multiple allosteric sites? And are you looking into it?
No. Yeah. So the short answer is yes, there can be, and that will make it more complicated. So actually, our thermodynamic cycle shows just coupling between two sites. But in reality, there could be multiple sites who are talking to each other. And that is where a more general Markov state model kind of thing will be required. And already we have seen, see, what I mentioned was all you have to do, we have to compare two conformational ensembles. One is with the apo and another is bound. Now, when you combine or compare, you will see that there can be differences in many different sites. And now, once you have identified these sites, one has to analyze that which of them is more effectively connected to your active site. So these are the more detailed analysis one has to do. But the first step would be to compare the conformational ensemble between these two, apo and bound. And then you can actually identify multiple sites. And it may even so happen that targeting two sites parallelly will be more effective, like having multiple molecules acting together in two different sites, and that may have more cooperative effect as inhibitors. But yeah, these are all very useful and interesting questions for future.
Sure. About the laches, the promiscuity of the laches. So, are they being used in industries?
Yes. Actually, this is well known. This laches as an enzyme is well known. And because this is a kind of it's promiscuous enzyme, it is used generically. But the mechanism, to the best of my knowledge, that how it actually works on this wide range of molecules, it was not studied. So that is what we have looked into. And in future, we also intend to look into the actual chemistry part of it. So far, we have focused actually on the binding. So actually, we have focused on the substrate binding, but we did not look into that actually how it actually donates electrons. There is a multi-copper site. That's where it's more complicated. It's a metalloenzyme. And in future, we intend to look into the chemistry of it. And that is where quantum chemistry has to be invoked. Yeah.
Yeah. Yeah. Okay. Since there are no more questions, again, I would like to thank Professor Chakrabarty for very nice talk. It was really interesting.
Thank you.
And I'm sure that our audience have thoroughly enjoyed it. Now I will hand over the stage to Well Kumar for vote of thanks. Well.
Hey, thanks, Anil. Hey, thank you, Dr. Suman. Really appreciate you taking time and spending the last 1 hour with us. So on behalf of everyone at Fusion Talks and at Pre-Science, I really would like to extend our heartfelt thanks for your enlightening session. Yeah, my name is Vel Kumar, so I manage the operations for Prescience. So your insights into the dynamic nature of proteins and their role in a biomolecular function were truly inspiring. So thank you for sharing your knowledge and expertise with us. So it was an absolute privilege to have you as a speaker. So thank you so much, sir.
Thank you for inviting me. It was a pleasure for me also. Yeah.
Appreciate it. Yeah, Anil, you can.
Yeah, so thank you, everyone. Yeah, thank you,
Thank
Dr.
you.
Suman Chakrabarty.
Bye.
Bye.
You all. Bye. Take
Bye.
care.
Bye.
Thank you. Bye.
Yeah.